is a professor and ex-head at Department of Pharmaceutical Technology, Jadavpur University. He has been affiliated with various other institutions, including Marie Curie Institute. He has been Marie Curie International Incoming Fellow, and he has been associated with Manchester Institute of Biotechnology at University of Manchester, as well as at Italy, IRCCS Instituto di Ricerche Farmacologiche Mary Negri. I hope I am pronouncing it correctly. In addition to that, he has been member of editorial advisory board of various prestigious journals, and he has been recipient of, as I told you, Marie Curie International Fellowship, Commonwealth Academic Staff Fellowship, AICTE Career Award, and DST Fast Track Scheme for Young Scientists. Today, he's going to present to us about the DTC laboratory, the Drug Theoretics and Cheminformatics laboratory, QSAR tools from Jadavpur University. So the stage is yours, Professor Roy.
Thank you, Dr. Bagler, for the nice introduction. And I also thank Dr. Raghava for inviting me to speak on this particular topic. So today I will give my speech on DTC laboratory QSAR tools from Jadavpur University. Jadavpur University is a state-funded university in the state of West Bengal. According to NIRF ranking, it is number 8 in the university ranking. I belong to Department of Pharmaceutical Technology, and in our lab, Drug Theoretics and Cheminformatics laboratory, we develop QSAR models with application in drug design and property modeling in material science, in toxicity modeling, with special reference to ecotoxicity modeling of different chemicals of emerging concern, and also for nanotoxicity modeling, modeling of antioxidant action, modeling of sweetening agents and flavoring agents, and several other endpoints. We are also involved in development of new descriptors for QSAR models, and we have developed a group of descriptors which are called ETA, which are involved, which are already incorporated in different software which are known worldwide, like Dragon, Alpha descriptor, Paddle descriptor, etc. And also we are involved in development of different tools for QSAR model validation. We have developed RM² metrics, which are used by different research groups. We have developed MA-based criteria. We have developed new criteria for applicability domain of QSAR models. We have developed different criteria for QSAR model validation. So I will now go to the topic. Before I go to our own tools, I will give a brief introduction to QSAR, because all participants may not be familiar with QSAR. QSAR is acronym of Quantitative Structure Activity Relationship. It is basically a statistical approach of correlating response property with chemical structure information. Now, the response property may be biological activity, but it may be toxicity or any other property of chemicals. And chemical structure information are encoded in the form of numerical values, which are called descriptors. So we develop statistical relationship between the response with chemical structure information. So the basic notion of any QSAR is that the response property is function of chemical structure or properties. And when the response property is in continuous scale, we can go ahead for regression-based modeling. And if the response is graded in nature, we will go for classification-based modeling. Now, from the pharmaceutical perspective, if we give some examples, say we have 3 drugs, chlorpromazine, promethazine, and ethopropazine. All are sharing the same scaffold, the phenothiazine nucleus, but they have different substituents and side chain. And according to those substituents and side chains, we have different pharmacological categories like antipsychotic, antihistaminic, and anti-Parkinsonian. So this small example gives us an idea how changes in substituent pattern can change the pharmacological category. It also can change the duration of action. It can change the type of action. etc. So this kind of relationship between molecular structure and changes in biological activity in a quantitative fashion is the center of focus of QSAR. But QSAR is not limited to application to biological activity itself. It can be applied to different property endpoints. So we can model different properties. For example, say in material science, if we give examples of polymers, they have different properties like say dielectric constant or the other important properties of polymers. So we can model those and we can derive or design suitable materials which will be of our use using their optimized property. It can be used for toxicity modeling. From ecotoxicity model point of view, we can model fate, environmental fate of chemicals of emerging concern, their biodegradation, etc. So as I mentioned that QSAR is a ligand-based approach, so we must have a series of compounds with known structures, and we should know their property or biological activity values known to us. And from the chemical structure, we should encode the information in that is present in those structures in the numerical form, which are called descriptors. Now we apply appropriate statistical tool to model the endpoint using statistical approaches. And finally, we get some pattern known to us, which can be used to design new compounds and which can also be used for predictions of a new set of compounds. So when we model property or toxicity, then we term QSAR as QSPR and QSTR. So we start with molecular structure representation. We calculate molecular descriptors. We apply appropriate statistics and fitting, and then we can predict properties and activities. So the QSAR table, a typical QSAR table looks like this, where we will have our endpoint that is being modeled. In case of QSAR, that is biological activity, and that is a therapeutic activity. But in case of QSPR or QSTR, that will be some target property or toxicity. And we'll have a series of columns containing the chemical information values, like say log P or pK, or different kind of computed descriptors. So there are different levels of theories for computation of descriptors. So we can compute descriptors using simple fingerprints, or we can calculate descriptors considering the 3D structure of molecules. So it depends that which kind of features we use for our modeling. Then we apply appropriate feature selection tool to know which features will be important for us for modeling purpose. And then we apply appropriate regression or classification-based strategies to develop the model. So once we get the model, we have to check the reliability of that model from the statistical sense, and also we have to judge their potential of application to new set of data. And for that, we need to apply different kind of statistical tools. And once our model is properly validated and we are confident about their applicability, we can predict a new set of compounds, or we can design new set of chemicals, maybe with better predicted pharmaceutical activity, or with less predicted toxicity. So QSAR has different steps, starting from data preparation, data processing, prediction, validation, and data interpretation. Usually for biological QSAR, we take logarithmic transformed form of the response, and then we prepare our QSAR table. And I have already mentioned that to check the predictive ability of a model is very important, and usually a separate set of data for checking the predictability of QSAR model may not be available. So what we do, we divide our available data set into two parts. One is called training set and another is called test set. The training set is used for model development, but test set is kept aside. Although we know the experimental values of the test set responses, but we never use this test set compounds. So these are actually unseen compounds to our model. So once the model development is complete, then we apply our model for prediction of the tested compounds. So tested compounds are unseen to model. So tested information has not been used in the learning process. So this can be used as a test how good or bad our model is in prediction of values for the new set of compounds. But in doing so, we must check the applicability domain of our model, because all QSAR models are developed based on similarity principle. So the tested compound should be close to the training compounds, at least to some of the training compounds, because if it is not close to the training compounds, then it may contain some features that the model has not seen. That means the model has not been trained for those kind of features. So model cannot respond well for those kind of features. So it will fail. So test set compound should be close in the chemical space to the training set compounds. And then we have to apply a series of metrics to know the statistical quality of the model, because all QSAR models are statistical in nature. So there will be the probability functions and the different kind of features, degree of freedom, etc. So that we can be sure that the statistical acceptability and robustness of the model is fine. This is apart from the usual test of validation to check the reliability of the model. And finally, the results will be interpreted from physiochemical sense so that we can apply the gained knowledge for design of new compounds. I've already mentioned that QSAR is applied not only in drug design, but in material science and predictive toxicology. And nowadays, the application of QSAR in predictive toxicology is very vast, especially for regulatory perspective. In case of ecotoxicity prediction for hazard identification, hazard categorization, and prioritization of chemicals, QSAR has broad applications. So for QSAR, we need some experimental data available, because without experimental data, we cannot develop any QSAR model. But we use experimental data for limited number of compounds, and then we develop our QSAR model, and then the developed model can be used for prediction of a large number of compounds, and probably it can be used for screening a large database also. So we are using experimental information from the limited number of compounds, and ultimately we are applying on a broad number of compounds or large database. So QSAR can be used for a filtering technique to prioritize the compounds for further synthesis. So in drug discovery, the experiment is unavoidable. Experiment will be there, but we need to do these experiments with a large number of compounds, because the probability of success in drug development is very low. And as you know, that the drug development process is very long and tedious, and there is involvement of a lot of manpower and money. So to work with the large number of compounds, to work with a large number of candidate molecules will have significant expenses. So we have to increase the probability of success. So to increase the likelihood to work with the potential compound, more potential compound, we have to apply some rational strategy, and QSAR can be that rational strategy. For example, say many compounds, even with good in vitro activity, may fail in vivo, and sometimes this kind of failure is visible at later stage of drug development, and sometimes maybe after marketing also, because we see the unforeseen side effects of many compounds at later stage, even after marketing, and then more delay we do in identifying some undesirable side effects, maybe some admit failure, etc. More cost involvement will be there. So we should identify such kind of failure at very early stage, probably before synthesis of a compound. So the admit testing should be done in silico, even before synthesis of a compound. So at that point of time, there is no experimental test possible, but we can apply different filtering tool strategies and admit model to filter out the compounds which may have some undesirable admit behavior. This is the drug development stages, discovery and development stages, preclinical research, then the investigational new drug application, then clinical research, phase 1, phase 2, phase 3, then comes the NDA application, and after that drug approval, and then also the phase 4 clinical trial continues. So it takes 12 to 15 years of time to see a compound in market. So it is a long journey, and still the probability of success is very low, 1 in 10,000. So we have to apply this kind of rational strategy to reduce the number of compounds that we have to use for our experiment purpose. And QSAR, along with some other related in silico tools, has been applied to find out useful drugs like tirofiban in thrombosis, imatinib in cancer, raltegravir in AIDS, donepezil in Alzheimer's disease, and so on. So these examples are from a pharmaceutical point of view, but as I have already mentioned, that QSAR has significant application in identification of chemical hazards. As you know, there are several hundred thousands of compounds in commercial use, but we have data for less than 1% compounds for their ecotoxicological hazard and risk. So, and it is not possible to gather data for thousands of different endpoints for thousands of compounds using experiments, and especially from the 3 R's concept, the animal experiments are discouraged. So the regulatory agencies are relying on the theoretical techniques. So US EPA and European Commission Scientific Committee on Toxicity, Ecotoxicity, and Environment are recommending QSAR as essential tools for ecotoxicological risk assessment. Not only toxicity, but also different property endpoints like environmental fate, ecological and health effect of organic chemicals in absence of experimental data can be predicted using QSAR. Especially the PBT chemicals, persistent, bioaccumulating, and toxic substances, they can be modeled. And we can go for green chemical design using QSAR. And this concept is also coming to pharmaceuticals, green pharmacy, that green pharmaceuticals design. Because pharmaceuticals are chemicals of emerging concern, not only pharmaceuticals, biocides, cosmetics, products, and different endocrine disruptors. So these are compounds of emerging concern, and we can apply QSAR to better design pharmaceuticals and cosmetics, etc., by tailoring and removing some features which are potential for their ecotoxicity. So QSAR can also be used for exploring mechanism of toxic action of chemicals. In view of rich regulation in the European Union, QSAR is being used to gap the data, to bridge the data gaps, and this can be used as an additional data with experimental data when submitting data to the REACH authority. So QSAR supports 3 R's principles, and these are supported by different regulatory organizations like ECVM, International Organization for Medical Sciences, US EPA, OECD, etc. And different endpoints for predictive ecotoxicology includes irritation, carcinogenicity, dermal toxicity, developmental toxicity. In material science, different materials like ionic liquids, polymers, nanoparticles, ceramics, etc., can be subjected to QSPR modeling. I've already mentioned about QSAR table, which looks like this. There will be 1 or more columns for your response, but there will be several columns for your descriptors. But all descriptors are not important for the particular response. And also there may be some redundant descriptors. There may be some intercorrelation among descriptors. There may be some descriptor columns which have the constant variance. And so this kind of descriptors should be omitted. So we should have some data curation tool to reduce the size of the QSAR table. And then we can apply appropriate feature selection tool to know which kind of features are important for our particular response, which may be activity or property or toxicity. And then we apply our regression or classification-based tool to develop our model. In case of regression-based tool, if we develop a good regression model, then if we plot experimental versus predicted or calculated value, then we'll see that Our plot will show less degree of scatter, so that will be close to the 45-degree angle line. So that is a good diagnostic measure from the scatter plot about the quality of the model. This is the historical evolution of QSAR model starting from 1868. It has come a long way from the time of Crum, Brown, and Fraser. At that time, physiological action of restricting derivatives or correlated with mathematically correlated with chemical constitution. Then Mayer-Wotton theory for narcotic potency with olive oil water partition coefficient. Then came the first QSPR model at the time of Hammett, which was actually applied in physical organic chemistry. Then the proper biological application was made at the time of Hunch in 1962. Then Free Wilson, the mathematical model. Then came graph theory, the application of different homologs of alkyl hydrocarbons with different degree of branching, etc. And from that, different connectivity indices evolved. Then came ecotopological state atom indices and Kappa-Sheva indices. Then came 3D QSAR in 1988 by Kramer. Then the COMFA, etc. And from 90s onwards, various ligand-based and structure-based approaches. And from 1997 onwards, multi-dimensional QSAR. So we see the dimensionality in QSAR, 0D is chemical formula derived, 1D substructural fragment derived, 2D graph theory derived, 3D spatial or geometry derived. In case of 4D, conformation, orientation, and protection steps incorporated. 5D induced fit derived. 6D is solvation condition derived, that is incorporated. And 7D, we use real target-based receptor model data. So QSAR is nowadays applied in pharmaceutical sciences along with other in silico techniques like ligand-based techniques, pharmacophore, and structure-based techniques like molecular docking, molecular dynamics, etc., and applied in virtual screening and for other cheminformatic applications. Now, coming back to the statistical techniques of QSAR, it is mainly of two types, regression-based and classification-based. The popular regression-based method include method of least squares, so that is multiple linear regression, and partial least squares, and classification-based method, discriminant analysis, logistic regression, etc. And with the development of machine learning approaches, we also use artificial neural network, support vector machine, decision tree, etc., for QSAR problems nowadays. Now, from the classical QSAR model development point of view, there are some requirements for the endpoint values. Usually for biological activities, we take a dose for response type of activity like IC50 and EC50, which should be expressed in molar unit. And this should be converted to the negative logarithm, that is log 1 by C. And there should be good span of 4 to 5 log units of the response data to get reliable model. And there should be sufficient number of data points. As I mentioned, QSAR is statistical approach, so number of data points should be higher and number of descriptors should be lower. The number of descriptors in the final model should be lower. And the typical multiple linear regression looks like this, where you can see that an equation like this. In the left-hand side, you have the response variable, which is the activity being modeled, and in the right-hand side, you have the independent variables, x1, x2, x3. These are basically descriptors. And you have the A1, A2, A3, these are the regression coefficients, which will be obtained from the regression problem. So once you get the regression coefficient values, you can calculate different metrics for judging the quality of QSAR models, like determination coefficients, explained variance, variance ratio, and standard error of estimate to check the statistical quality of QSAR model. But these metrics have nothing to do with the reliability of your model on the new set of data, because these are completely based on the fitting techniques. Now, in case of classification-based models, we have different series of metrics like sensitivity, specificity, accuracy, and precision, which are obtained from the contingency table. And we can also have different other derived measures. I'm not going into details. Now, QSAR model development and validation. are done according to OECD principles. There are 5 guidelines. There should be a defined endpoint, an unambiguous algorithm, a defined domain of applicability, appropriate measures of goodness of fit, and mechanistic interpretation. So we can actually show the different steps of QSAR model development according to OECD principles, like the defined endpoint for OECD principle 1, then this descriptor generation and division into training and test set, and then application of appropriate statistical tool, and according to OECD principle 2. According to OECD principle 3, there should be appropriate application of applicability domain. According to OECD principle 4, there should be application of different measures of goodness of fit and validation measures. And according to OECD principle 5, there should be mechanistic interpretation of the model. Now we should remember different points for QSAR model development, like data homogeneity, inappropriate endpoint data, collinear descriptors, incomprehensible descriptors, error in descriptor values, transferability of QSAR. Now, these are very important. When a QSAR model is developed, it should be reproducible. That means if some other group wants to reproduce the model, so one should be able to develop the same model by applying this same methodology. So one should enlist all the steps very accurately, and one should also take care regarding the error in descriptor values, because it may so happen one is using some software for descriptor calculation, and that particular software may not be able to handle some kind of feature. Say some particular metal is present, because most of the QSAR descriptor computing software do not consider metals. Some software may not be able to consider, say, iodine. So this kind of features should be remembered during descriptor computation. And there should be appropriate applicability domain measurement. Generation of data points should be avoided. Use of inadequate data. It cannot be done with very small data set. Then replication of compounds in the data set. When one is using more than one data sets or databases, the same compound may be present in different names. So one should take care the same compound should not be replicated. The narrow range of endpoint values, the endpoint values should not be too much a narrow range, because if there is not sufficient variation in the endpoint values, then the statistical model cannot be developed. Then overfitting of data should be avoided by using limited number of descriptors. Lack of inadequate statistics and misinterpretation of statistics, because most of the cases QSAR is applied by non-statisticians, so it should be properly handled. And inadequate training test division, failure to validate QSAR correctly, mechanistic interpretation, etc. So these are the points one should remember. Now, regarding validation of QSAR model, as I've already mentioned that QSAR model development from statistical point of view, that is more like a fitting technique, but it has nothing to do with the application on the new set of data. So we have to apply on the new set of data to know and to understand whether it is applicable for the new data. And for that, we should apply different validation measures, either using internal validation or using external validation. In case of internal validation, we use the same set of data for model development by using methods like leave one out or leave many out. In case of leave one out, we omit one compound in cycle, and from the reduced data set, we regenerate the model, and using that regenerated model, we predict the omitted compound. And in this way, in different iteration, we omit one compound, and this will go on. The number of iteration will be as many as the number of compounds present in the data set. In case of leave many out, we actually omit a certain percentage of compounds from the data sets, and one can understand that this will go in several iterations, because there are several combinations possible to omit a certain percentage of compounds from the data set. In case of external validation, some new set of compounds are used for validation purpose. Usually what we do, we divide our data set into two parts, training and test. That is one way of external validation, but this is not strictly external validation. If we have some other new set of compounds for external validation, that is the gold standard of application of QSAR model. Apart from that, one can apply bootstrapping, which is a kind of internal validation, and also one can apply wide randomization, where the Y column is randomized and new models are developed. And the random model should have the low statistical values than the non-random model. And one can also perform the X randomization also in some cases. So I will not go into details here because we have limited time. So there are some strategies for selection of training set components. As I've mentioned that we have to apply the similarity-based measure, because all QSAR model based on similarity-based measure. So we can apply statistical molecular design or self-organizing map, k-means clustering, k-means, etc. We can sometimes use the sorted response also for division of the data set into training and test. One more important thing is that all QSAR models are not applicable for all type of query compounds. There should be an applicability domain test, because the query compound may not be similar to the training compounds based on which the model has been developed. So there should be some degree of similarity, and usually the applicability domain is measured by leverage approach, which is obtained from hat matrix. And there is a great degree of debate for internal versus external validation also. We will not go into that debate here because we perform both internal and external validation. And that should be done. So now we come to our tools. So our tools are available in the shown websites. In the websites, we have developed a series of tools, and there are some small tools also which are required for QSAR model development, sometimes a division of training and test, or some detect curation, or say leave many out validation. For those, we have some small tools also. But for in general, for QSAR model development and validation, we have a series of tools. There are some tools that just I'm mentioning, but I'm not going into details. I'll go into details of some specific tools like MLR plus validation, then dataset division, GUI, the external validation plus for calculating different external validation metrics like Q square external F1, F2, F3, RM square, CCC, and so on. These are mainly for regression-based models. Then the base subset selection, genetic algorithm for MLR model development, bias variance estimator, etc. Now we'll go into details on some specific tools. The first one is the applicability domain standardization approach. I have mentioned that the query compound should be within the applicability domain, and they should be close to the training compound in the chemical space. This work was published in Chemometrics and Intelligent Laboratory Systems in 2015. So here we consider that the training compounds are actually normally distributed both with respect to the response and also the descriptor values. And as we know, the mean plus minus 3 times standard deviation covers 99.7% of the population. So if some query compound is within this range of the training data for the corresponding descriptors, then obviously this will be within the general pool, and there should be some similarity to the training compound. But it is beyond that. That means this is somewhat different from the training compounds. So there is some question whether that particular query compound is usable for prediction purpose by our model. So here we use an algorithm. So a QSAR model will have a number of descriptors. So for each descriptor, we calculate the standardized value, that is the descriptor value minus mean of that column divided by standard deviation, the usual measure of standardized value. So we calculate the standardized descriptor value for all descriptors present for both training and test compounds. And then we take the minimum value of the standardized one. And if this value is less than 3, then obviously the compounds are within range. Because if the minimum is less than 3, then for all the descriptors, the value will be within 3. So that means there is no outlier in the training set and no compound outside applicability domain in the test set. And if this is for maximum value, if the maximum value is less than 3, and if the minimum value is more than 3, that means all the compounds for all descriptors, the value will be more than 3. In that case, that particular compound will be outside the applicability domain in case of test set, or in the training set that will be an outlier. But the question comes when the minimum value is more than 3, but the maximum value is less than 3. In that case, we use a standard score based on Z score, and we calculate this S new value, that is the mean of those values plus 1.28 times standard deviation. And if this S max value is more than 3, in that case, we consider that that particular compound is outside domain, and it is less than 3, then with 90% probability that is within the domain. The next tool is double cross-validation. This is applicable to remove the bias in descriptor selection, because in case of training set, we have a fixed composition of chemical compounds. So when we apply some feature selection tool, there will be some biasness in selection of descriptors. So instead of using the same training set, what we do, we divide the training set several times into a calibration and validation set. So this work was published in Chemometrics and Intelligent Laboratory System in 2016. So we do K-fold cross-validation, and from K calibration set, we derive K models, and K models are applied on K validation sets. And then on from the performance of the validation set in terms of say least MAE, mean absolute error, or we may use some consensus prediction also using the top models, or by finding the best descriptor combination from the top model, we can select the best model. So instead of using a model that is obtained from fixed composition of the training set, we can use this method. That this is more preferred than using the simple training test division. Next one is the intelligent consensus prediction. Now, multiple models are more preferred than single model, because it increases the applicability domain of the models, and also it can nullify some shortcoming of a particular model. This was published in Journal of Chemometrics in, I think, 2017. So here we are using intelligent consensus. That is not simple average. This comes from the concept of some qualified individual model that comes from that the concept of distance of the query compound from the close compounds in the training set. We take most close 10 compounds, but while considering this closeness, we also consider the distances among the training set components, and based on that we select the qualified model. So we do not take all the models, we take average of only the qualified models, or we can take the weighted average predictions also in CM2, or best selection of prediction in CM3, and we have additional criteria based on Euclidean distance cutoff, applicability domain, and Dixon Q test. And finally, we have seen from different studies that CM2 and CM3 perform better than the other consensus model and also the individual models. And the fourth one is prediction reliability indicator. This is applicable for MLR and PLS models. This was published in ACS Omega in 2018. And this is based on 3 criteria. First one is scoring based on mean absolute error of closest 10 training compound for each query compound. In criteria 2, we consider the scoring based on applicability domain. And in criteria 3, we consider scoring based on proximity of the observed response mean to the proximity to the observed response mean of the training set. And based on these 3 scores, we give a composite scores, and based on that, we classify predictions as good prediction, moderate predictions, and poor or unreliable predictions. Finally, I will talk on QSAR modeling of small data sets, as it is customary to divide the data set into training and test. In case of small data set, it becomes difficult for us to divide into training and test, because we are going to lose significant amount of chemical information, and we are not using those compounds for modeling purpose. So model may not be that much robust. So in that case, what we should do? So we can apply this double cross validation, which actually, instead of dividing the data set into training and test, we can use double cross validation, which stratifies the training set into calibration and validation set several times. And also we can use consensus model. And as I've already mentioned, in case of consensus model, we can use different combination of descriptors In case of a particular model, we can use only a limited set of descriptors, but in case of consensus model, without sacrificing the statistical quality, we can use multiple descriptors, and we can expand the applicability domain. And also we should take care of chemical curation, because it may so happen the structure we are using is wrong, normalization of bonds is incomplete, some duplicates are present, other possibilities are wrong charges, presence of explicit hydrogens in hydrogen duplicate structure. One example I can use, keto-enol tautomerism. So one compound may be present in keto and enol form. So depending on the keto and enol form, it will have different descriptor values, especially for topological ones. And also, so for nitro group, that nitro group can be presented using different representations. So the calculated descriptors may be of different value depending on the type of representation. So one should go for chemical curation. So we have a NIME workflow here, which has element filter, connectivity filter, addicate molecules, addicate salt stripper, the optimization of geometry, and structure normalization. And finally, we have the SDF writer. And when we obtain this ultimate the SDF file obtained, then we can apply small dataset curator, which can do duplicate analysis, structural outliers removal, response outlier removal, and activity cliff analysis. In case of duplicate analysis, the duplicate analysis is done based on calculated descriptor values. Outlier analysis based on the descriptor values, that is simply based on mean plus minus k times standard deviation, and also based on response value, we can find a response outlier. And one more important thing is activity cliff. Two compounds which are very close in the structures, but they have different kind of activities. They may not be modelable using QSAR model, so it's better to remove those compounds. So we have different strategies based on the number of compounds, number of such compounds available. So this is there in our small dataset curator tool. I'm not going into details here. And and we have a small dataset modeler, where instead of dividing the dataset into training and test, we use the whole set and we go for double cross validation with exhaustive lead layout, and there will be several combinations, and we can generate the MLR and PLS model using GA technique, and finally find out the best model based on their performance on the validation set using several criteria. So this is our small dataset modeler, and this is the graphical user interface for our small dataset curator and small dataset modeler. And this work was published in Journal of Chemical Information Modeling in 2019, I hope so. And this is the more recent, the DTC QSAR, that actually clubs the different small tools into one, which can handle both regression and classification-based QSAR models, and it can handle descriptor selection using genetic algorithm and also base subset selection. And it can also give results for random forests, etc. And it has some graphical output also. And more recently, we have developed a read across tool. Read across tool is a similarity-based measure. It is more applicable for toxicity estimation in case the limited number of compounds are available. And it works using three kinds of similarity measures: Euclidean distance-based measure, Gaussian kernel-based, and Laplacian-based. And based on some kind of weightage, we can predict the toxicity or activity of the query compound based on the similar compounds present in the training set. And all this, okay, so for these tools, if there's a form in our website, if anybody is having any kind of problem during using these tools, then he can ask questions using this form. We can reply to her or him immediately. And these tools are available here in these two particular sites. And we also acknowledge funding received from UGC, ICMI, ICT, and DBT different time period. And most of the software programs were developed by Provi Nambure, which is presently with ProtoQSAR in Spain. These are some text for beginners of QSAR. So understanding basic of QSAR, and this is a more brief form in Springer VIP for QSAR modeling, and there are some more advanced reference for QSAR. And these are more recent application of QSAR in different fields like computational modeling of Alzheimer's disease, multi-target drug design using cheminformatic approaches, in silico drug design for drug repurposing, ecotoxicological QSAR, and the most recent in silico modeling of drugs against coronaviruses. And the forthcoming one, chemometrics and cheminformatics in aquatic toxicology. So this, my presentation, there is some question. I'll be happy to take those questions. Thank you.